welcome to class to Chapter 3 on nucleotides, nucleic acids, and genetic information. In this chapter, we're going to split it up into two different sections that each talk about a little bit of a different topic on nucleic acids. The first section, which we'll talk about in this video, is about nucleic acid structure and function. In this part, we'll talk about nucleotides and the components that go into them, as well as how those contribute to making up both DNA and RNA. In our next video, we'll talk a little bit more about some nucleic acid techniques, how we work with nucleic acids in the lab. So, one important part of looking at the structure of nucleic acids as we break them down is that each part of a nucleotide contains a nitrogenous base. And the nitrogenous bases help us differentiate some of the different nucleotides from one another. So the first base we have on the left is called a purine. So purines have a two-ring structure. Each of these rings contains nitrogens. And you can see the general structure of a purine present both in the two examples of nucleic acid nitrogenous bases. On the top we have adenine, which is sometimes abbreviated A. Adenine has that basic purine structure except with two differences. So we have an amino group here at the top, and then at the bottom, instead of that bond to hydrogen, we have a bond to X, or an R group, which in this case is a sugar. The other purine nitrogenous base is called guanine. So guanine differentiates from adenine in a couple of different places. First at the top here, instead of an amine group, we have a carbonyl. And then on the side over here, we have an amine group where adenine does not have a functional group. And again, we have this bond to sugar. On the right, we have the pyrimidine bases. So pyrimidines, there are three different ones that we see in most cellular genetic material. The first one is cytosine. Cytosine is found in both DNA and RNA. So cytosine, like the other bases, is attached to a sugar where it says X over here. There is a carbonyl group here that differs from our sort of generic pyrimidine structure at the bottom of the slide. And then on the top of the ring, cytosine has an amine group. The other two pyrimidine bases are called uracil and thymine. So in the middle here we have uracil. Uracil is only found in RNA. So again, we have this link to a sugar at the bottom, but what differentiates uracil from cytosine is at the top here we have a carbonyl group. Our last pyrimidine base is called thymine, and thymine is only found in DNA. And you can even tell that from this diagram because in this link to sugar it says little dx, and the D stands for deoxy, which is what differentiates the sugar in DNA from the sugar in RNA. And what differentiates thymine from uracil is that in addition to the other functional groups, it also contains a methyl group here. So next we're going to consider how these nitrogenous bases bond to each other. As a lot of you probably already know, DNA is always a double-stranded molecule, and what holds together the two strands are hydrogen bonding between the types of nitrogenous bases. So at the top we're showing the bond between adenine and thymine. Adenine always bonds to thymine with two hydrogen bonds, which you can see outlined here. And then on the bottom, we have an example of a guanine, And in addition to thymine, in some cases, adenine is also able to bind to uracil, because as we recall from the last slide, the structure of uracil is very, very similar to the structure of thymine. On the bottom, we have an example of guanine hydrogen bonding to cytosine. Here we can see that these two bases bond to each other with three different hydrogen bonds. So guanine always binds to cytosine, and they always use three different hydrogen bonds to do that. So what is the next part of the structure that defines a nucleic acid? So here we'll consider nucleosides. The definition of a nucleoside is it contains a nitrogenous base, like one of those last 5 we considered on the last couple of slides, in addition to a sugar. So on this slide we have pictures of the 2 main sugars that we see in the nucleic acid structures in our cells. On the left we have ribose, and on the right we have deoxyribose. These are both 5-carbon sugars, and the carbons in each of these molecules is labeled by number. So the first carbon is here, second carbon is here, third carbon is here, fourth is here, and the fifth is up here. The main structural difference between ribose and deoxyribose is on the second carbon, carbon number 2. In ribose, carbon number 2 has a hydroxyl group attached. In deoxyribose, as you might guess from the words deoxy, that there is no oxygen there. There is only a hydrocarbon there, the carbon number 2 bound to hydrogen. So how can we form nucleosides in combination with all of those 5 different nitrogenous bases we learned about? On the bottom of the slide we have a list of all the names we can have. So we have our nitrogenous base adenine. When it's a nucleoside, when you have adenine attached to, for example, a ribose, you'd call that adenosine. If you have adenine attached to deoxyribose, you would call that molecule deoxyadenosine. And the name change from adenine to adenosine helps you identify just from the word that it is a nucleoside. So you have a base attached to a sugar covalently. Similarly, guanine, when it's in the nucleoside form, it's guanosine or deoxyguanosine. Cytosine becomes cytosine or deoxycytidine. And in the case of only RNA, as we mentioned before, uracil only is part of RNA, so there is no deoxy form when it's attached to a sugar, so you only have uridine, which is uracil attached to ribose. There is no such thing as a uracil attached to deoxyribose, at least in nature. And then similarly but different, we have our thymine. When it's in the nucleoside form, it's only called deoxythymidine. Because that thymine is only found in DNA, you're only going to really see it on a deoxyribose. So when you have the nucleoside with the deoxyribose attached to thymine, you have deoxythymidine. The next type of nucleic acid structure to consider is the nucleotide. So before we had a nucleoside, that was sugar plus base. Now a nucleotide, we are adding on. So now we have the base plus the sugar plus at least 1 phosphate. And generally you will see anywhere from 1 to 3 phosphates attached. So on the left side in this figure, part A, you have a ribose, again with that 2' OH group. On the right side you have a deoxyribose. So on the left side, our ribose is found in RNA. On the right side, our deoxyribose is found in DNA. We have our nitrogenous base attached to both of them. And then these are drawn with phosphate groups attached at different carbon positions. So on the left, our ribose, we have a phosphate group attached at the 5' carbon. So this is what you'd call a 5' ribonucleotide, 5' telling us where the phosphate is attached, ribo telling us that our sugar is ribose, and the nucleotide because you also have that nitrogenous base involved. So you have all 3 of those components, the phosphate, the sugar, and the base. On the right, you have our piece of a DNA nucleotide, and here the difference from the one on the left is our phosphate is now attached on the 3' carbon of that sugar. But again, you have a phosphate, a sugar, and a nitrogenous base. You have a nucleotide. This is a 3' deoxynucleotide. So nucleotides, the ones I showed on the last slide as an example, had just 1 phosphate group attached. So when you're going to name them, you can name them depending on how many phosphates they have. So I mentioned how you can have anywhere from 1 to 3 phosphates. So if you were to name your nucleotide, you would name it first with the nucleoside's name. So for example, adenosine, and then how many phosphates. If there's one phosphate, you could call it adenosine monophosphate. If there are two phosphates or a diphosphate ester attached here, you can call it a nucleoside diphosphate. And if you have three phosphate groups, then you would call it a nucleoside triphosphate. So an example given on the right side of this slide is a nucleoside triphosphate. And this one has adenine as the nitrogenous base. So we have our sugar is ribose because our 2' carbon has a hydroxyl group attached. And you have three different phosphate groups here. So now we know we can name this molecule adenosine because adenine plus sugar, tri because there's three phosphates, phosphate, adenine triphosphate, otherwise known as ATP, which you've probably heard of. It's used as the energy currency in the cell. And additionally, it's a component when we're making up our nucleic acids like DNA and RNA. So when we're talking about DNA and RNA, in nature we usually see them as a polymer, a string of nucleotides, or a polynucleotide, which we call a nucleic acid. So all nucleic acids contain a sugar phosphate backbone, meaning it has just what we have been talking about. You have our phosphate groups alternating with our sugars going down the molecule. And the phosphate groups, usually when we are considering a polymer like this, the sort of last phosphate is on the 5' end because it's attached to that 5' carbon. If you follow that molecule down, you'll have the phosphate group on the 5' carbon, and then you'll have the sugar, the rest of the sugar, then the 3' sugar attaches itself to the next phosphate group, which is then attached to the 5' carbon of the next sugar, and so on and so forth. So you can sort of think of this molecule as having a directionality, where we're thinking about it going from 5' to 3'. And usually we'll see the last phosphate on the 5' end, and on the 3' end, the last sugar, the 3' carbon, usually is just depicted with a hydroxyl group. It's not depicted with the next phosphate because of sort of how these molecules are found in nature. That's typically what we would see. And so because of that directionality, we can start thinking about writing our DNA and RNA sequences with that same directionality. And we usually think of it with that 5' to 3' direction. So, and you can start naming your structures just usually based on the nitrogenous base, because that is what differentiates your nucleotides. So for our example on the top, we have an adenine, followed by a uracil, followed by a cytosine, followed by a guanine. So if we were to write the sequence of these 4 nucleic acids as a polymer, you could write 5' end, and then you have A, U, C, and G, followed by the 3' end. And that will always inform whoever you're giving this information to which direction your sequence goes in. And how do you know if the molecule we're looking at right now is DNA or RNA? Once again, that brings us back to our sugar. If we look at the 2' carbon, if it is attached to a hydroxyl group, like we see here, we have ribose. So we know that this is actually going to be a strand of RNA. The other thing that gives away the idea that this is actually RNA is we have our uracil base, which is, again, usually only commonly found in RNA in nature. And finally, for DNA at least, you often have a complementary sequence because DNA is often found as a double-stranded molecule. And we can always figure out the complementary sequence to any strand of DNA because of those known hydrogen bondings that we talked about between the nitrogenous bases. So our adenine at the top will always form 2 hydrogen bonds to thymine. Our uracil, which in the case of DNA would actually be a thymine, so that's why they drew this little methyl here. So you'd have a T here if it was DNA. And that would form 2 hydrogen bonds with another adenine, or with an adenine. Our cytosine would form 3 hydrogen bonds with a guanine, and our guanine would form 3 hydrogen bonds with the cytosine. And with DNA, the polymers are always formed in the anti-parallel direction, meaning that if the one that's drawn here on the left goes from 5' towards 3', the one on the right, the complement, would actually go in the 5' to 3' going up the slide. So 5' down here, 3' up there. And that has to do with, the reason for that is that that's how the nitrogenous bases can form those hydrogen bonds with one another. One will always be essentially flipped 180 degrees in order to form the best spatial distance for hydrogen bonding. So that brings us to the general nature of a DNA polymer. So we talked a little bit about how you have two strands that come together and they stick together through those hydrogen bonds. But when you have long enough strands, they actually form this double helix shape that you see on the right. And again, that's helped along because of this hydrogen bonding. The hydrogen bonding, the 2 between adenine and thymine, and the 3 between guanine and cytosine, help the DNA molecules to form a particular spatial distance from one another, and when they curl around each other, they form a very stable structure. So you can see it's drawn here in green on these sort of ribbons on the side. That's the sugar phosphate backbone. And then in the middle, sticking out, you can see the nitrogenous bases. So for example, this one towards the top, you have the C cytosine here, triple hydrogen bonded to a guanine here, and in the dotted lines in between them, you can actually see the 3 hydrogen bonds. Similarly, if we look about halfway down the molecule, we can see an adenine here, and that has 2 hydrogen bonds that help link it to this thymine on the other side. So one convenient thing about DNA is because we know about the way that adenines always bond to thymine and cytosines always bond to guanines, we can know how much of each base we have in any particular DNA molecule, as long as we know how much we have of one particular base. And this is called Chargaff's rule for double-stranded DNA. And so the Chargaff's rule states that the number of purine bases will always equal the number of pyrimidine bases. And we can sort of reason that through if we sort of look again at the way that these bases bond to each other. So adenine, which is a purine, will always bond to thymine, the pyrimidine. Similarly, guanine, the purine, will always bond to cytosine, a pyrimidine. And purines always pair up with pyrimidines similarly because of this particular spacing inside the double helix. You have a certain distance there that is always taken up by a pyrimidine plus a purine going horizontally. So that is why Chargaff's rule makes sense. So the percentage of purines always equals the number of pyrimidines. The percentage of adenines always equals the percentage of thymines, because again, they're bonded to each other. If you have one, you have the other. And same thing, percentage of C always equals percentage of G, because if you have one, you will always have one of the other, because they pair up. And you also know that the total number of bases in the molecule is 100%. So if you know, for example, how many C's you have, you can figure out how many G's, because it's the same number. And you know that the rest of them are A's and T's, which are equal numbers to one another. So finally, once you've gotten all that DNA in that double helix, how do we actually manage to cram all that genetic information into the nucleus of the cell? So our genome contains all of the DNA within our whole organism, and it is a lot of DNA. It's usually measured in kilobase pairs, or thousands of base pairs, each base pair referring to one nitrogenous base. And in the case of humans, our genome contains billions of base pairs, which is a lot of base pairs. So we had to find a way to essentially organize up all of that DNA and also keep it easy to find when we want it, and also have it take up very little room, which is how we come up with chromosomes. So chromosomes are found in each one of our cells, and they contain a number of different DNA molecules. In the case of humans, we are diploid, which means that we have 2 copies of each chromosome. So in almost all human cells, we have 46 chromosomes, which means we have 2 times 23 chromosomes, 23 being our haploid number. So on the right in this picture, we see our autosomes, of which we have 22 pairs, so it's chromosome 1 through 22, and we have 2 of each. And then we also have our sex chromosomes, and each cell contains 1 pair of those, either XY or YY, with some rare exceptions. So that being said, when we think about the structure of DNA as that double helix, when it's all stored up, squished together in that chromosome, when our cells are ready to divide, they need to be able to replicate that DNA, to have 1 copy in one daughter cell, 1 copy in the other daughter cell. So the structure of DNA itself acts as a template for when that genome is replicated. Because those base pairs always dictate what the other strand is going to be, we can separate the 2 strands of the double helix apart, and then each one has directions for how to make a brand new strand, which you can see being built along here. So the enzyme that helps make that happen is called DNA polymerase, and DNA polymerase essentially reads the template strand and helps attach on a new complementary nucleotide, and that is how new DNA molecules are built. So each DNA double helix always consists of 1 DNA strand that's sort of the original, or what's written here as the old, and then 1 newly synthesized strand that's made as an exact anti-parallel copy of the old one. And you can see how that works, because T is always paired to A, G is always paired with C. That's all you need, is just 1 strand to act as a template. So that's DNA. Now let's talk about our other nucleic acid, RNA. RNA has a number of different functions that all act to help express our genes. So the first one, the one that a lot of people most commonly think of when they think of RNA, is called mRNA. The M stands for messenger RNA. So messenger RNA carries genetic information out of the nucleus. It literally provides a message from our nucleus to the cytosol of our cell, where the ribosome can then read off that mRNA to make a protein. rRNA is another important kind of RNA, where the little r stands for ribosomal. This is ribosomal RNA, so it's found in ribosomes, and that rRNA is really helpful for when we have to synthesize proteins. It helps to stabilize the ribosome, as well as to act as an enzyme and help catalyze some of those reactions that are happening when we're making RNA into protein. The third type of RNA that is really important genetically is tRNA, and where the T is for transfer. So this transfer RNA helps to ensure the correct sequence of amino acid within a protein. And the T refers to transfer because it helps to literally transfer on the amino acids when the protein is being built up. So when we think about other functional types of RNA, aside from those main 3, first we have to consider ribozymes. Ribozymes are RNA. They're structurally made of RNA, but they act as enzymes, so they help to catalyze chemical reactions. Specifically, as you can tell from the name here, ribo, it's found in the ribosome. So this is some of that rRNA that's found in the ribosome, and it has a really important function to actually help build proteins. Another type of important RNA that we use in the lab particularly is called siRNA. siRNA has the function of regulating gene expression. It's able to bind to an RNA template and actually prevent it from getting translated. So it has a sort of opposing function to the other types of RNA that we might see in the cell. And finally, as a sort of interesting fact, we also know that some viruses actually use RNA as their genetic material instead of DNA, like organisms, including humans, do, and most other larger organisms do. There are some reasons why this is helpful for viruses. In some ways, it's really good for organisms that replicate really quickly, because you can sort of skip a step in replication. You can just go from RNA straight to protein. You don't have to go through the trouble of making DNA into RNA into protein. But this could be really disadvantageous for an organism like a human, because human cells replicate relatively slowly, and RNA tends to overall be much less stable than DNA. So if we were to use RNA as our genetic material, we could expect to see just issues with being able to store that genetic information throughout the lifetime of the cell or the organism, and that could cause a problem for gene expression and for replication. So we've talked a little bit about the 3D structures of DNA, that double helix and those chromosomes. Now we can think a little bit more about the structures that RNA likes to make. So a single strand of RNA, which you see here, is sort of what we would traditionally think of in mRNA as having that appearance, just a single strand of RNA. But any RNA as a single strand is able to form all kinds of 3D structures. A really common one is depicted here, called a stem loop structure. So on the stem portion, we can see hydrogen bonding between complementary bases, and that's why you have a stem. The strands of the RNA are drawn close to each other. On the top portion, we have a loop. The loop was where we have sort of incompatible mismatching RNAs. They are not going to try to hydrogen bond with each other. And so you end up with a sort of ballooning out almost. That looks kind of like a loop. So that's what we call it, a stem loop structure. And this can help to give large, long strands of RNA a big 3D structure. So in the middle here, the example that we see is in a tRNA. So tRNAs are always folded like this. There are 3 stem loops shown in green, blue, and red. And then in yellow here at the top is actually where the amino acid is going to be attached for when that tRNA helps to start build that protein out of individual amino acids. And then at the bottom part of the tRNA, shown in gray here, is the part of the tRNA that recognizes the template, the message from the messenger RNA, to help match it with the correct amino acid. And when you think of a tRNA as a 3-dimensional structure, it actually looks almost like an inverted letter L. And that's the same, it's colored. These 2 diagrams have the same color scheme, and you can see here at the top, this yellow part is again where the amino acid attaches. So finally on the right, we have the structure of a ribosome, the 2 large main subunits of the ribosomes. The 2 main subunits of the ribosome. The top we have our large subunit, and the bottom we have our small subunit. And you can see mixed in together, you have these purple structures and these orangey structures where the purple is the RNA, the rRNA, and the purple is the protein and the orange is the rRNAs. So that sort of brings us to a concept we've been discussing a little bit called the central dogma of biology. So the central dogma of biology pretty straightforwardly states that when we have DNA as our genetic material, it can be made into RNA, and then that RNA can be made into protein, and that protein carries out some kind of function within the cell. So we've already discussed a little bit about how not all RNAs are made into proteins. Some of them, like our RNAs and tRNAs, don't get translated, but a lot of them do. So that's sort of, we can really kind of think of this as for mRNA. So, and the processes for each of these arrows is sort of shown on the right. So for DNA, as shown on the top in blue, it's made into mRNA. And the process of doing that, using DNA, the template to make mRNA, is called transcription. And the enzyme that we need to help us catalyze that reaction is called RNA polymerase. mRNA has these sets of 3 nucleic acid bases that we call codons. And those act as little templates for tRNAs. tRNAs, the gray part in that diagram I showed earlier, recognize that piece of the mRNA through the same idea where nitrogenous bases pair with one another, and then the amino acid attached to that yellow part of the tRNA that we showed before then will get added onto a growing protein in a process called translation. And the enzyme that catalyzes that reaction is called a ribosome. So all in all, the central dogma of biology sort of has an explanation of how the genetic information in our cells goes through a number of different steps, and eventually gets made into a protein that helps the cell to function. So we talked a lot about transcription and translation and how those are sort of related to the different nucleic acids in our cells. They can be very kind of fun to watch, kind of fascinating. There's a lot of different moving parts and pieces. So I'm posting links to these 2 videos here where you can see both transcription and translation in action. They're kind of interesting to watch. If you want to give them a watch, I'll let you do that on your own time. So now with that, you should have learned enough from this lecture to be able to go ahead and answer the Chapter 3 Nucleic Acid Structure and Function practice questions from the document I posted on Brightspace. Thank you.